Hello, my name is Daniel Azar, and today I will present to you my recent paper in Structure Cell Press titled Vaccinia virus immunomodulator A46 Destructive Interactions with MAL and MyD88 shown by negative stain electron microscopy. This study was realized together with colleagues from Tim Skuren's lab at the Max Perutz Labs, Medical University of Vienna, Vienna Biocenter, Austria, and collaborators from Boštjan Kobe's lab at the University of Queensland, Brisbane, Australia. Toll-like receptors initiate the innate immune responses against infectious organisms. They signal through their tier domains by forming open-ended assemblies with the downstream adapters. These adapters, namely MAL, MyD88, TRAM and TRIF, are inhibited by Vaccinia virus A46, a member of the poxviral BCL2-like protein family. However, the mechanism of interaction of A46 with its targets had remained unclear to this point. Ve et al. NSMB 2017 showed that the tier domains of MAL and MyD88 form filamentous assemblies in vitro and protofilamentous cooperative assemblies in cells, leading to the subsequent recruitment of signaling molecules in the TLR signaling pathways. In this study, we show that A46 destroys MAL and MyD88 tier domain assemblies in a concentration-dependent manner by targeting their intra-strand interfaces, and that the multiple interactive facets of A46 include its alpha-7 helix and C-terminal flexible region. In order to unravel the elusive and destructive protein interactions in question, we used negative stain EM and protein-protein cross-linking. First, we showed that full-length A46 is required to inhibit the in vitro MAL and MAL-induced MyD88 tier domain assemblies, whereas its separate domains showed less or no effect. Second, by performing serial dilutions of A46 and acquiring microscopy and protein turbidity data, we showed that the destruction of both adapter tier domain assemblies by A46 is concentration dependent. In the case of MAL, we observed a gradual increase of the filament diameters in addition to their gradual destruction, to the point of having no surviving assemblies at an equimolar concentration of A46, as is quantified in panel I. In the case of MyD88, the filament-based crystals become gradually more brittle and their filament striations more irregular, to the point of having no surviving assemblies at an equimolar concentration of A46. Based on previous interaction data and on our knowledge of the A46 structure, we performed mutational analysis of residues on helices alpha-1 and alpha-7 of full-length A46. We demonstrated that all our alpha-1 mutants showed an attenuated or abolished effect on MAL assemblies, whereas neither one showed a different phenotype than wild-type A46 on MyD88 assemblies. On the other hand, both our alpha-7 mutants showed an abolished effect on MAL assemblies, whereas one showed an attenuated phenotype on MyD88 assemblies. Additionally, we cross-linked A46 to both MAL and MyD88 and found that A46 interacts with sites mainly involved in forming the intra-strand interfaces in both signalosomes, and that its C-terminal flexible region appears to play an important role in both cases. Based on these findings, we proposed a model in which A46 targets the MAL and MyD88 intra-strand interfaces, first through an interface penetration by the C-terminal flexible region, and through gradual incorporation leading to late-stage complete assembly destruction of both MAL and MyD88.